welcome back to our channel. Today, we're diving into the fascinating world of epigenetics. You may have heard that your genes play a significant role in your health, but did you know that your behaviors and environment can also have a profound impact on your genetic expression? Join us as we explore epigenetics and its implication in health and diseases. Epigenetics is the study of how our behaviors and environment can cause changes that affect the way our genes function. Unlike genetic changes, epigenetic changes are reversible to a certain extent and don't alter our DNA sequence except in the later stages of a disease, as they influence how our bodies read that DNA sequence. Before we delve deeper into epigenetics, let's clarify the concept of gene expression. Gene expression refers to how often or when proteins are created from the instructions within our genes. Genetic changes can alter which protein is made, while epigenetic changes affect gene expression, turning genes on and off. Turning them on will result in the production of a certain molecule or a signal which can result in a particular disease. Keeping them off often results in the status quo of health, and hence the importance of understanding the epigenetic switch. So, how exactly does epigenetics work? Let's explore the three main types of epigenetic changes. 1. DNA methylation. DNA methylation involves adding a chemical group to DNA at specific places, blocking the proteins that read the gene. This process turns genes off. Conversely, demethylation removes the chemical group, turning genes on. 2. Histone modification. DNA wraps around proteins called histones. When histones are tightly packed, proteins that read the gene have limited access to the DNA, turning the gene off. Conversely, when histones are loosely packed, more DNA is exposed, allowing the gene to be accessed and turned on. Chemical groups can be added or removed from histones to regulate gene expression. 3. Non-coding RNA. Non-coding RNA helps control gene expression by attaching to coding RNA, breaking it down so that it can't make proteins. Additionally, non-coding RNA can recruit proteins to modify histones and turn genes on or off. They are often the signal to stop the genetic or epigenetic modification. Our epigenetics change as we age, as a part of normal development and aging, and in response to our behaviors and environment. Let's explore three key aspects. 1. Epigenetics and development. Epigenetic changes begin even before we are born. All our cells have the same genes, but they look and act differently. Epigenetics plays a crucial role in determining a cell's function, such as whether it becomes a heart cell, nerve cell, or skin cell. For instance, consider the difference between a nerve cell and a muscle cell. Both have the same DNA, but epigenetics allows the muscle cell to activate genes essential for its function and deactivate genes relevant to a nerve cell's function. 2. Epigenetics and age. Our epigenetics continue to change throughout our lives. For example, a study compared DNA methylation in a newborn, a 26-year-old, and a 103-year-old. It revealed that DNA methylation levels decrease with age, with newborns having the highest and centenarians having the lowest levels. Prevention of DNA methylation prevents aging and increases the quality of life. 3. Epigenetics and reversibility. Not all epigenetic changes are permanent. Some can be added or removed in response to changes in behavior or environment. Take the example of smokers versus non-smokers versus former smokers. Smoking can result in specific epigenetic changes, but after quitting, former smokers can gradually regain normal methylation levels provided they have not reached a point of no return like a squamous cell carcinoma of the lungs. Epigenetic changes can profoundly impact our health. Let's look at three ways in which this happens. 1. Infections. Germs can manipulate our epigenetics to weaken our immune system, aiding their survival. For instance, Mycobacterium tuberculosis causes tuberculosis and can change histones in immune cells, weakening the immune response. Certain mutations increase cancer risk, and some epigenetic changes can do the same. For example, abnormal DNA methylation levels near specific genes can affect gene expression and raise the risk of certain cancers, which are termed as epigenetically modifiable cancers. A pregnant woman's behaviors and environment, such as her diet, can influence her baby's epigenetics. These changes may persist for decades and make the child more susceptible to certain diseases later in life. The increase in the prevalence of type 2 diabetes in babies born with mothers with gestational diabetes is a very real scenario which is unfolding in front of us. Almost all non-communicable diseases which have great implications in the current healthcare systems like coronary artery disease, stroke, chronic renal disease, diabetes mellitus, hypertension and its complications and a variety of cancers have a definite epigenetic switch which when switched on results in all the disastrous complications, long-term morbidities and higher death rates in this subset of patients. And there you have it, a brief overview of epigenetics and how it influences our health. Our behaviors and environment have a profound impact on our genes, and understanding epigenetics can help us take control of our health in new and exciting ways. Almost all the modern non-communicable diseases like obesity, diabetes, hypertension, cardiac diseases and certain cancers have an epigenetic component needs to be understood and studied for better outcomes and reversal of these diseases. Make sure to subscribe to our channel for more updates on groundbreaking scientific discoveries. Thanks for watching and stay healthy. See you next time.